everyone, so in this video I will be talking about enzymes. So this mainly focus for students who take O-level combined science and also biology, but again there is no limit to it, so anyone can watch this video. Alright, so let's get started. So what is enzymes? So enzymes, for non-science term, okay, for a non-scientific term, I can say enzyme is like a scissor, okay? It's like a scissor. So I draw a small scissor here, showing that enzymes, its job is to cut, okay? To cut bigger things into smaller things, okay? Just for example, you cut A4 paper, okay? A4 paper into smaller pieces of paper. So that is enzymes, okay? But inside your body, of course, it has lots of job, okay? But basically it's just cutting, right? So first let's get to know about enzyme. Enzyme is our actor, okay, in this video. So we need to know our actor. So what is enzyme? The first one is very popular description. So enzyme is a biological catalyst, okay? So enzyme is a biological catalyst, and maybe you heard catalyst from chemistry. So what is catalyst? It speeds up a reaction, okay? So biological here means that this catalyst, okay, can be found inside a living organism, not only human, but enzyme you can find also in plants, okay? Plants, animals, bacteria also have enzyme. So that is the first one. It is a biological catalyst. So catalyst, if you know catalyst, it speeds up a reaction. So you can say enzyme is a biological catalyst that speeds up a reaction. That is the first point. Second one, okay, what is enzyme? It is made up of protein, okay? Enzyme is made up of protein, okay? Enzyme is made up of protein. But this is biological catalyst, so this enzyme can be found inside your body, okay? When it can be found inside your body, most of the time it is made up of protein, okay? You are also made up of protein. Your hair, your skin, your muscles, they are all protein. So enzyme that can be found inside your body is also made up of protein, okay? Because they are made up of protein, they are very sensitive to temperature and pH. I will explain later, alright? So first one, it is a biological catalyst, it speeds up a reaction, and the second one is it is made up of protein, so they are very sensitive to temperature and pH. So that is the second one. Third one is they are specific, okay? They are very specific. What does it mean? It means that enzyme, for example here, this is enzyme. Enzyme has active site. This is an active site. Active site is the site which is active. So it means substrate will bind to this active site. So from this active site, the substrate will break into two pieces or how many pieces, okay? And that is the products. But they will only bind to a specific one, which means, I will explain later, that is the lock and key theory. According to the shape, it will start to break the substrate down. For instance here, of course, from the shape here, you can see that this enzyme will only break this substrate. Then you have the products, okay? That is why I say enzyme is very specific. So what happens after it binds to the substrate? Will it change? Okay, that is the fourth point. Enzyme will not change itself. So enzyme remain unchanged, okay? Remain unchanged. So even after this broken down process or breaking down process, this enzyme will not change its active site. It will release the product, then it will bind to another substrate and then break it down again, then bind to another substrate and break it down again. So this shape will maintain as long as these two doesn't affect, these two will not affect this active site, okay? So again, what are the 4 points about our actor today? The enzyme, right? So enzyme is a biological catalyst, so it speeds up a reaction. Okay, it speeds up a reaction, and this enzyme can be found inside your body. So that is why it is made up of protein. And also it is very specific, so it will only break down specific substrate. It will not break other substrates. So for instance, if this is amylase, it will only break down carbohydrates. If this is protease, it will only break down protein, like that. Then the fourth one, after the process, this enzyme will not change itself, so it remain unchanged after the process. So in exam, you are asked, what is enzyme, or define enzyme, and the question is 2 marks question. So what should you answer? Just take 2 points, okay? Just take 2 points. You can say enzyme is a biological catalyst and it is made up of protein. You have 2 marks. If the question asks for 3, give 3. If 4, then give 4. Okay? But usually it's 2 marks question. If 3 marks question, usually they will ask like define amylase, or what is amylase? That is usually 3 marks. First one, you can take this one. Amylase is a biological catalyst and it is made up of protein. The third mark, it has to be specific for the molecules that it will break down. So the third point should be amylase will break down carbohydrates to glucose. That is your third marks. Alright, so we cover this one, the biography of our actor in this video. Second one is the lock and key theory. The lock and key theory is very popular inside your textbook or your notebook. And maybe your teacher keeps saying lock and key theory. So what is lock, what is key? Imagine this enzyme is the lock. This enzyme is the lock which has a specific shape to a key. And the substrate is the key. Lock means kunci mangga that always pagar your gate. So you lock your gate or you lock your house with the kunci mangga, right? So the lock. So that lock is the enzyme and key is the substrate. From here, I have this shape. Okay, I explained this earlier already, so you might have the answer already. So from these 3 substrate, which one will bind to this enzyme? Okay, or from these 3 key, okay, among these 3 keys, which key will unlock the lock? Is it the round green one? Is it the triangle blue one? Or the black one? Of course, from the shape you see, this substrate will fit the active side of the enzyme. So what happened is, now you have enzyme substrate complex. So enzyme substrate complex. Not complex as in difficult, complex as in they become one whole unit. So this is called enzyme substrate complex. Right. So what happened when they bind? Of course, this enzyme will break the substrate. How it breaks? Okay, it breaks and give a products. Imagine this enzyme break this substrate into 2. So you have, let's say it breaks at this point, okay? It breaks at this point. So you have one small square and also one big square. Then the active site is now free, okay, or vacant. So another substrate which is of a similar shape can go in again, and it will be broken down by the enzyme, and then you have products. So it is repeated, right? So you have to understand this, because I will talk about the example here. Again, enzymes are very specific, so it will only break substrate. Okay, it will only break substrate which has the same shape as its active site. What happened when this enzyme found this the substrate that has its shape? It will collide or bind and form enzyme substrate complex. So enzyme substrate complex. what happens when they bind? This enzyme will break down the substrate into pieces, into smaller pieces. Imagine it breaks this into two, so you have these two shapes, and these are called products. So that is the lock and key theory. It talks about the specificity. What happened to the enzyme after the process? It will remain unchanged. So it will break down another substrates of the same shape. Okay, so this done. So let's talk about example. These three examples are very popular in exam. In fact, in your notebook, okay, your teacher also teach you this. So the first one, you have a molecule and it is made up of the same monomer. Okay, single molecule there, single particles there, okay, joined together and form larger molecule. So what is this? That is your, what should I write? That is your carbohydrates. Carbohydrates, or it is also called starch. Okay, carbohydrates, starch, your bread, your rice, potato, they are carbohydrates, or they starch is also examples of carbohydrates. So carbohydrates or starch are broken down by starch or carbohydrates. They are broken down by an enzyme called amylase, right? So I taught combined science students, O-level students, and most of them, they do not know that amylase break down carbohydrate. They always remember these two enzymes, but not amylase, PT amylase. So now you should know that carbohydrates is broken down by amylase. So keep telling yourself that carbohydrate is broken down by amylase. Carbohydrate is broken down by amylase. Starch is broken down by amylase. Starch broken down by amylase, right? So carbohydrates or starch broken down by amylase into smaller products. Okay, so now you have 6, 1, 2, 3, 4, 5, 6. So now the black chain is broken down. Okay, it's cut. Okay, like I say, enzyme is like a scissor. Okay, so it is broken down, cut. So you have what is the product? The product is called glucose. Okay, so sometimes this part, first example, carbohydrates broken down by amylase into glucose, they can connect this question to respiration, because glucose is very important in respiration, because in respiration glucose react with oxygen to give carbon dioxide, water, and also energy. So without glucose, no respiration can happen, so you will become weak. Okay, respiration, even now I'm talking here, my body is respiring. Okay, it used the food, okay, food that I've eaten just now, and convert it to energy. That's why I have the ability to record this video, because I got my energy from the food I eat. So enzyme is working inside my body so that it can produce energy for me to speak here. So that is the relation, right? So that is carbohydrates. Carbohydrates broken down by amylase, and the products will be glucose. Carbohydrates or starch broken down by amylase into glucose. Right, so let's move to the second one. Second one is similar like number one, but it has different monomer. There's red, blue. Of course, you cannot find the blue, red, green inside your body. This is just a simple diagram. So if it is made up of different monomer, this is protein. Okay, this is protein. This is protein, because protein, okay, protein is made up of amino acid. There are different types of amino acid. So protein is made up of different types of amino acid. So if I say that protein is made up of amino acid, then you will know what is the product. So here, protein. What is the enzyme? Easy. This is, I think, the most popular enzyme that the students know. So every time I ask my student, carbohydrate is broken down by which enzyme? They will say this enzyme, protease. Okay, so the enzyme is protease. So when I ask, fats is broken down by which enzyme? They will answer protease. So protease is the most popular one, but in exam, it's not really popular. The most popular one is amylase. So please remember, carbohydrate and starch is broken down by amylase to glucose, right? So here protein is broken down by enzyme called protease. You see there, pro. So pro is for pro. Okay, protein is broken down by protease into amino acid. So you have triangle, then you have this red circle, and green. Okay. So you have amino acids, single molecules. So they are not bonded anymore. Okay, it's protease cut the bond. Okay, cut the bond and release the monomer. So monomer is the single molecule that make up the larger molecule. So these are the monomer. So amino acid, what is it used for? Amino acid will be used by your body, your organs, the cells, okay, to make a protein. So it will go back to your cells and make a protein. So you have a muscular body, or your hair also, okay, your skin. That is the function of amino acid. So what is this monomer called? It is called amino acid. Okay. So carbohydrate and starch broken down by amylase into glucose. Protein broken down by protease into amino acid. So the last category is called fats. Right? This is fats. So what is the enzyme that break down fats? So this is a bit difficult to remember because the enzyme, the name of the enzyme doesn't really relate to the name fats. But if you know another name of fats, okay, another name of fats is actually called lipid. Okay, so if you know lipid is fats and fat is lipid, you will know that the enzyme is called lipase. Lip. Okay, so lipase break down fats into one glycerol and 3 fatty acids. Okay, so 3 fatty acids and 1 glycerol. Okay, so fats is broken down by lipase into 1 glycerol and 3 fatty acid. The chain, okay, the chain here got broken down. Okay, is cut by the lipase. So you have 1 glycerol and 3 fatty acid. So these are the product. Again, carbohydrates and starch broken down by amylase into glucose. So if you are asked what is the enzyme that break down carbohydrate or break down starch, you say amylase. What is the enzyme that break down protein to amino acid? You say protease, pro. Okay, and then what is the enzyme that break down fats into 1 glycerol and 3 fatty acid? It is lipase. Right, good. Good. Of course, good. So you should understand that one, because this one and this one will be asked in one question, like in this experiment. We will discuss this one after this one. Okay, so sensitivity. Like I said, enzyme is made up of protein. We are also made up of protein, so we are very sensitive to temperature, and we are also very sensitive to pH, to acidic, to alkaline. You will have sensitive skin if your, let's say if your shower gel, okay, your shower gel, the pH, your skin cannot tolerate the pH, then you will have itchiness and whatnot. So first we go to temperature. So temperature, I put here 37 degree because that is your core body temperature. Your normal body temperature is around 36 to 37. So 37 is the optimum temperature. So what happened at low temperature? So just imagine yourself is, just imagine enzyme is like yourself. Imagine outside is raining, okay, outside is raining, and then you are in this room with aircon on. It's cold and you feel very comfy to just sleep on your bed without doing anything, so you just want to sleep and become inactive. Same goes to enzyme. Enzyme at low temperature, the enzyme activity will be less because no energy. And again, temperature, whenever physics, chemistry, or biology, when they talk about temperature, it means they are talking about kinetic energy. Remember this, if in exam they talk about temperature, high temperature, low temperature, and then they ask about the particle, it means they are talking or they are asking about the kinetic energy. What is kinetic energy? Movement energy. Enzyme is moving, okay, but because low temperature, they're not moving, they just want to sleep, okay, they become inactive. Like you don't want to do anything during rainy day. Same like enzyme. So at low temperature, let's say 0 degree, 20 degree, like that, but it slowly goes up as the temperature increase from 0 to maybe 20 to 25, like that, it started to go up. Of course, so the higher the temperature, then there will be increase in enzyme substrate complex. There will be increase in the formation of enzyme substrate complex, because enzymes start to move now and look for the substrate and then bind and then give out products. So the activity will increase as the temperature increase. So at 37 degree, that is the optimum temperature, which means that is the best temperature for the enzyme to work. So it will take in substrate, break it down, take it another one, then break it down, so you have lots of product. So that is the optimum temperature. But beyond that 38, 39, 40, it starts to go down. So it means less enzyme substrate complex. Why? So if lower temperature, if in exam if the question asks why at low temperature the formation of enzyme substrate complex is less, you can answer because the enzyme is inactive. When inactive, it means the enzyme just sleep, but the active side still the same, it will not change. But higher temperature, At higher temperature, the enzyme substrate complex started to decline, started to become less as the temperature goes beyond 37. Why? Because the enzyme here, the active site, like you, okay, like you, if you go out and work under the sun, the hot sun, under the scorching hot sun, what will you say to yourself? I'm melting. Same, enzyme is also melting. That's why I say you see enzyme like yourself. Enzyme is also melting. So, okay, maybe I just use this one. So, imagine this is the active site at temperature beyond 37, it starts to melt and change its shape. Maybe the shape change and become like this. So this is called, what is this called? Denatured. In exam, if you are asked what happened to the enzyme beyond 37 degrees Celsius or at 40 degrees Celsius, any temperature beyond, okay, over 37 degree, you answer the enzyme, the active site of the enzyme got denatured. This is the word that the examiner wants. You have to answer this one, denature. What is denature? Denature is when the active site of the enzyme change. It will not go back to the normal shape. So this one cannot be used anymore and it will be broken down then by, don't worry, your body will keep producing enzyme. So now, can this substrate enter and bind to the active site? No, it cannot bind because the shape of the active site has changed. Therefore, there will be no enzyme substrate complex. If there is no enzyme substrate complex, can you have products? No. That is why you have to keep your body at optimum temperature. When you feel cold, try to bring your body to up to this temperature. But of course, your body will do it by itself. And also, when you feel hot, especially in fever, when you have fever, usually you'll be given porridge because it is easy to digest. Correct? Why? Why you cannot eat heavy food when you have fever? Logic. Your body now is not 37 degree. You have high fever. When you have high fever means temperature inside your body is high. So when high, what happened to the enzyme? I said, denature. The active site change its shape. So let's say the burger you eat when you have fever, the burger is, let's say the bread, okay, is carbohydrates and starch. And the meat is protein. If denature, starch, carbohydrate, protein cannot be broken down by amylase or by protease. So what happened is your food there still bigger molecule. Eventually you will have indigestion. That's why when you have fever, you eat food that can be easily digested. Alright, so that is temperature. And again, that is why when you have fever, you have to eat or consume medicine to lower down your fever. That is the reason why. So your body can operate like normal. So you can eat your burger, your chicken wing or whatever it is, because your body now at 37 degrees. So remember, temperature, at lower temperature, enzyme become inactive. Okay, become inactive. At higher temperature, beyond 37 degree, what happened? The enzyme denatured. What does denature mean? Denatured means the active site of the enzyme has changed its shape, so substrate cannot bind to the active site. So there will be no enzyme substrate complex form, therefore no products. Okay, you have to relate everything. but it should be easy lah. Don't worry. Next one is pH. So I have 3 environment here. 2, pH 2 is acidic. That is your stomach. Your stomach is the most acidic place, okay, the most acidic place inside your body, because it contains hydrochloric acid. So that is pH 2, very, very acidic. 7 is neutral. Neutral, so it means your saliva. Your saliva is pH 7, around that pH. And usually, the enzyme that can be found inside your mouth is amylase. pH 2 is pepsin. That is actually member of protease. Protease have different enzyme also. We go pepsin and then trypsin. Trypsin is works well at pH 9, a bit alkaline, because it is produced by pancreas. Then it drains the pancreatic juice into the small intestine for further digestion. So we look at 2 first. In your stomach, okay, in your stomach, you can find same the temperature. The shape of the graph is always the same. You have like this. So imagine this is enzyme pepsin. Pepsin is actually a member of protease. Okay, so pepsin also break down protein, but because it works in acidic condition in the stomach, they give name so they don't get confused. So pepsin works at pH 2, but pepsin breaks down protein, same like protease. So pepsin break down protein into amino acid. So it works well at pH 2. Remember this, pH 2, pepsin works at pH 2, and this usually happen inside your stomach. So in case MCQ asks you in stomach what enzyme is working, okay, which enzyme works inside the stomach to break down protein to amino acid, so your answer is pepsin. Then the next one is pH 7, that is your saliva. So inside your saliva you can find amylase. So what enzyme? Amylase. So you can find in your saliva. Not only in your saliva, pancreas also produce amylase together with the trypsin. So amylase break down carbohydrates to glucose. That's why whenever you think about food, okay, especially at school, when your teacher teach in front of the class, but inside your head you are thinking about wings, about foods, okay, about foods, then suddenly your saliva starts to come out. Okay, that is the effect, because your brain thought that you are eating. Okay, your brain thought that you are eating, so it starts to produce saliva just to get ready for digestion. In your saliva, it contains amylase, so this will break down carbohydrates into glucose. So that is pH 7. Amylase work at pH 7 and can be found in your saliva or your pancreatic juice. So just right here, plus pancreatic juice. Next one, green. 9, that is a bit alkaline. Same. So at 9 is the optimum pH. Okay, if this is optimum temperature, that the peak is optimum pH. So this one, the enzyme is called trypsin in your pancreatic juice, right? So in your pancreatic juice, so it works at pH 9, and this can be found in your small intestine, the first part of your small intestine. Now for combined science, you might not know the trypsin and pepsin. Maybe you are told about protease only, but to be specific, pepsin breaks down protein also into amino acid, but in acidic condition. Trypsin, you see sin, okay, so trypsin is also a protease. It breaks down protein to amino acid, also same. But where it can be found in the pancreatic juice, released or produced by the pancreas. That is the difference. But they break down the same thing. Only the condition is different. Alright? So that is temperature and pH. And pH at lower temperature and at lower pH and higher pH. Example, this one. If lower than 2 or higher than 2, this pepsin, what happened? It will denature. For pH, if it goes lower or beyond the optimum pH, it will get denature. Simple. Not inactive, but it will get denature. Imagine you will have skin erosion, okay? Skin erosion when you take, or when your body bath or shower gel, the pH is not good for your skin. Or maybe acid, the strong acid, if you spill on your skin, what happened? There will be burn. There will be burn, so you will have that scar forever and ever and ever. Same like this one. Once it's changed, because enzyme is protein, once it changed the active site, it cannot change to normal. Same like your skin. If acid spilled on your skin, you will have that burn and it will remain there forever unless you do plastic surgery. Right. So below or above the optimum pH, just remember there will be denature. Same like this. Nice. Now the last one, experiment. So usually in paper 2, student always skip this question, and I don't know why. Well, I was a student before, and of course I skipped this one because it looks scary. You don't want to think about the question, so you find the easy question. But actually, after my O-level, I realized that this type of question, it is very easy to get marks. So try and read the question, because the answers are from this. Usually they will ask the definition of enzyme first before they go to the experiment. Then they will talk about temperature. You see, they put different temperature. So if you know the optimum temperature is 37, then you know which amylase is active there. And also pH depend on the enzyme. Like here it says amylase, so you know amylase works best at pH 7. That's it. If you know amylase work at pH 7 and at 37 degrees Celsius, you can already answer this question. Full mark for you, right? So this usually related to food test also, because you have starch solution and then you will be asked about iodine solution. This one simple. You just need to know iodine solution. What is the color? It is brown. Iodine solution is brown. But if starch is present, yep. If starch is present, I do positive. If starch is present, then it will change from brown to blue-black. Okay, brown to blue-black if there is starch. So this one also you need to know. Usually the most popular one is amylase and starch. Then they give different pH and different temperature. So let's go one by one. Maybe the question will be, there are 3 test tubes labeled A, B, and C. It contains starch solution and also amylase. Then after 10 minutes, that is too long, after 3 minutes or 1 minute, okay, after 3 minutes lah. After 3 minutes, sample from this solution will be taken, or iodine solution will be added to each test tube. So you need to investigate what happened to these 3 test tubes. First, test tube A. It contains starch solution and amylase. Inside your head now, amylase, starch solution. So amylase break down starch to glucose, right? So that is the first thing you should think. Then you go to pH. So you can see ngam, alright? Good. So starch and amylase is good. Amylase break down starch into glucose. Alright, so see the environment and condition. So first one is pH 7. So amylase, now you think, amylase works best at pH 7. So that one is also correct lah. But how about the second factor? It is 40 degrees Celsius. So this solution is heated up to 40 degrees Celsius. Then you go here. You have to think about the temperature, effect of temperature. The optimum temperature is 37. So it is beyond 37, 40 degrees. So what will happen to the enzyme, to the amylase when it is 40 degrees? The enzyme will be denatured. Why denatured? Because of the hot temperature, this active site will change its shape. What happens when it changes its shape? Substrate cannot bind to the active site. So there will be no enzyme substrate complex, therefore no product. So you just put cross here. So if these two is not thick, then it means amylase cannot work. Both must work. Okay, both must be at ideal condition and temperature. But this one only the pH is okay, but the temperature no. So the enzyme will still get denatured. So amylase get denatured. So what happened to starch? Will it be broken down? No. Okay. So here you can say 40 degree. It means denatured. Of course in exam you don't write this one, you write in full sentence. You say the temperature is 40 degrees Celsius, so the active site will be denatured because of the 40 degrees Celsius. Then what happened to the amylase? It cannot break down amylase, amylase cannot break down starch to glucose. So what happened? Then another question. Iodine solution is added to test tube A. What will happen to the iodine solution? So because starch is present here, okay, starch is present here because amylase cannot break down starch into glucose. So starch is there. So when you add iodine, if starch is present, what happens? It will change color from brown to blue-black. So iodine change from brown to blue-black. Alright, so because of 40 degrees Celsius, the enzyme got denatured. Therefore, amylase cannot break down starch into glucose. Therefore, starch is still present in this test tube. So, what if you add iodine solution? The iodine solution will change from brown to blue-black because starch is present. Right? So, that is the answer for A. How about B? So, we use red. How about B? Again, amylase and starch. Amylase break down starch into glucose. Alright, so again, let's check the parameters or the condition or the environment. pH 7. Amylase work best at pH 7, so that one is a tick. And then 37 degree. Oh, amylase work at 37 degree. That is the optimum temperature. So that is the point where it becomes active and binds to substrate and form lots of enzyme substrate complex and produce more products. So this one also thick. Both are thick, so this is an ideal condition for amylase. What happened? more enzyme substrate complex form. So what happened again? Amylase break down starch to glucose. So because of the two ideal condition, amylase now can break down starch into glucose inside test tube B. So what happened when you add iodine? In this test tube B, because starch is broken down into glucose already, so there will be no, so negative, I put negative for starch, so there will be no starch present in test tube B because all of it has been broken down by amylase into glucose. So what you have here is glucose. So iodine will only detect starch. If there is no starch there, what happened to iodine? It will remain brown. Right? So it will remain brown. It will not change to blue-black because all the starch inside the solution has been broken down by the amylase. So you can only see glucose there. Alright, the last one. Same, starch and amylase. Again, starch amylase, yes, break down starch into glucose. See the parameter again, it's pH 2. Oh, amylase doesn't work at pH 2, only pepsin is. So, amylase work at pH 7, so this is wrong. So automatically, the other one is correct, but only one is in ideal condition, which is 37 degree, which means amylase, yes, it works fine at 37 degree, but the pH is too acidic for amylase, so still it will get denatured. Like I say, if it goes lower or beyond the pH, optimum pH, it will get denatured. So here, same like here, but only change the 40 degree, pH 2, too acidic, amylase, denatured. So what happened? No starch to glucose. If starch is not broken down into glucose by amylase, so what happened? Starch is still present here. Starch is still present there because the enzyme got denatured, cannot break down starch into glucose. So if you add iodine, what happened? It will change brown to blue-black. Alright? So that is experiment. That is how you answer experimental question. If you understand this, then you can answer this one by one. Don't get stressed with the, I know if experimental question, they always have story, long story there, like wow, in far away kingdom, like that lah, similar like that. But just have a read, scan through, then you see the experiment, you see the parameter. If you know, then you can answer. You will get full marks, trust me. And the answer is simple. No need to give a very good sentence. They need the point. Okay, they need points. So you give the points to them. Use the proper words. Denatured. Okay, don't say melt. Okay, melt is only just for your understanding, but denature is the word. All right. So this is enzyme. This is the life of enzyme. So again, this is the biography. Enzyme is a biological catalyst that speeds up a reaction. It is made up of protein. That's why they are very sensitive to temperature and also pH. And they're very specific, so they will only break down specific substrate. So one enzyme will break specific substrate, like amylase is an enzyme that breaks down carbohydrates to glucose. Protease is an enzyme that breaks down protein to amino acid. Lipase break down fats only into 1-glycerol and 3 fatty acid. And then it remain unchanged after the reaction, so when after it breaks down one substrate, it will go and find another substrate, and then they combine again, then produce more substrate. That is how enzyme work. So the lock and key theory, that is the specificity, and then the sensitivity, just remember at body temperature it is 37 degree. Plants have a bit lower, I think 28. If the experiment here talking about plant, okay, about plant, usually the temperature they will put a bit lower, like 28 degree. So that's why you need to read the question. Temperature, if it is low, okay, lower temperature, enzyme become inactive. But as it goes up, as the temperature increase, go to 37, there will be more enzyme substrate complex form. That is the optimum temperature, 37 degree. Then beyond 37 degree, there will be less enzyme substrate complex form. Why? Because the active site gets denatured. So the substrate can no longer bind to the active site because the active site has changed. Okay, and this one cannot be reversed back to its original shape. So this one is no longer used. It is denatured. Denatured means active site has changed. Same goes to pH. Different enzyme have different pH. Like pH 2, pepsin works best at pH 2, and that one is in your stomach. So low pH, lower than 2 or above 2, pepsin will get denatured. Same amylase work best at pH 7. So below 7 or above 7, amylase will get denatured. And that can be found inside your saliva. And this one, trypsin, is also a protease. So pepsin is also a protease. But depending on the location, they have different names. So trypsin can be found inside your pancreas or in the pancreatic juice. So work best at alkaline environment like pH 9. So below 9 or above 9, they will not work because they get denatured. Therefore, less enzyme substrate complex form, less product. Here, the experiments, again, you just need to know all of this. Then you will be okay for experimental question. Again, look one by one. And remember, this one also is very important because sometimes they will change this to, let's say I put protease. If I put this protease, you see here pH 7, and then 37. Okay, I put this one 2 lah. Okay, 2, let's say. That is pepsin. Okay, that is pepsin. Sometimes they will put protease, but it's okay. Sometimes in the question they will say protease from the stomach, so you know it's pepsin. So pH 2 and 37 degree, starch solution and protease. Don't automatically you say, this 2 is correct, so it will become glucose. No, you see this enzyme. Alright, that's why you need to look one by one. Protease does not break down starch. Okay, protease break down protein. So the answer here will be wrong. The answer here will change. Alright, it will no longer this one, and also the iodine, the solution also change. So again, it depends on the experiment question. Okay, the question of the experiment. So I will write it back here. Okay, so this is pH 7. Okay, again, read the experimental question properly and remember this: amylase break down carbohydrate or starch into glucose, and protease break down protein into amino acid. Lipase break down fat into 1 glycerol and 3 fatty acid. Okay, so that's it for enzymes. So in the next video, I'm not sure which video I want to record, but hopefully this enzyme can help you understand your O-level syllabus or topics, right? So thank you for watching and enjoy.